Welcome to Bioinformatics with BB channel. My name is Babajan Bananganapulli. I'm an associate professor in bioinformatics. In this channel, we will cover various biological data analysis concepts in genomics, transcriptomics, proteomics, and metabolomics. If you like this content, please subscribe to our channel. Bioinformatics with BB channel reached 2000 subscribers within a month. I want to give you a big thank you for watching and subscribing to the channel. Last video we learned about a molecular docking of a protein-ligand molecule using Autodock. So in this video we'll look on to how we can analyze or interpret the docking results. This step is very important and very crucial if you are going for publication or if you want to publish your data in highly reputed journals. Okay, so here what the major things we are going to cover in this video. We are going to cover about how we can retrieve the Autodock results. Okay, so we are going to see about the DLG file analysis. We are going to see about how we can save the protein ligand complex in from the Autodock, and how to convert the protein ligand PDBQT to PDB format file. So along with that one, we also going to see about how we can visualize the protein ligand molecule in 3D and also 2D format. Let's start. If you recall previous video, the main objective of molecular docking is to attain the ligand receptor complex with optimized conformation and with the intention of processing a less binding energy. Okay, so the net predicted binding energy from the Autodock reveal the terms of various parameters such as hydrogen bonds, electrostatic interaction, torsion free energies, and dispersion and repulsion values like van der Waals interactions, dissolution energy, total intramolecular energy, and unbound system energy. Okay, so let's check .dlg file, docking log file, which we have generated from the Autodock, what the information is presented inside that file. Okay, this information is very helpful for us. We have to create a table like this. You can see here the table for your docking analysis. We have to use the RMSD values and also the binding energy, very important one, and also the inhibition constant. This table I got it from some publication. So one of our publication. So we added these values here. Okay, these values in the form of a table we added. We took binding free energies and also the inhibition constant, and this number of hydrogen bond amino acid interaction we'll see in the visualization part. Okay, so these are the two important things we have to consider, or we have to retrieve from the .dlg format file. So just let us check the .dlg format file. So this .dlg format file, we will get it from the docking results. Just type RMSD table. Okay, so click on RMSD table. So if you see here RMSD table, now it will show you various classes of information is there inside this table. Okay, so here is a ranking pattern. What is a ranking pattern is there? What is the sub ranks are there? In which run the binding energy has been you received, and what is the cluster RMSD is there, reference RMSD, all this information you will get this from this values. So already I told you what is the binding free energy. It's a combination of various things like hydrogen bonds, electrostatic, torsion free energy, dispersion, repulsion energy, and total intermolecular. Combination of all these things you will get this binding free energy of a ligand protein molecule. So if you see this cluster RMSD. Cluster RMSD is a root mean square difference. Root mean square difference in coordinate between the docking conformation and cluster reference. So the reference RMS is root mean square difference between the docked complex and the input structure. So here inhibition constant, if you see the inhibition constant, that is also very important one. If you scroll down to the little bit down, you will find the complete information about each and every round. You can see here the cluster 1 rank 1. This ninth run showed the highest rank out of the all the 10 runs that has been done by the Autodock. So you can see here the estimated free binding energy. As I told you here, it's a combination of various things like torsion free energies, unbound system energy, and different type of non-covalent interactions like van der Waals, hydrogen bond, dissolution energy. So combines together, you will find the free energy values. Along with that, you also find this inhibition estimated inhibition constant. So this Ki or inhibition constant is correlated to the half of maximal inhibitory constant at which 50% of the protein is inhibited. Okay, so this quantity measures indicate the amount of specific drug or substance need to inhibit the biological process. Okay, so the inhibition constant is a very important score to consider that can be measured the effective of compound as a drug. But you have to remember, so we are doing here a theoretical approach. We are just predicting this one. You cannot judge based upon this theoretical value the drug likeness of your compound. So you have to do, it's mandatory for us to come to the conclusion about the drug by doing the experimental studies only. So what I'm going to keep in this table, so I'm going to keep this reference RMSD. Okay, so here the reference RMSD of this structure is 0.85. Okay, so I'm going to keep this 0.85 here, and the binding free energy minus 14.17 kilocalories per mole, and also the inhibition constant I'm going to add 41.06 picomolar. So these are the three major things I'm going to consider from this table. Okay, so this number of hydrogen bonds and also the amino acid interaction we are going to see in the visualization part. Apart from various scores in docking file, the visualization of receptor ligand geometries and their interaction will give us better understanding in drug development procedure. So it's very important for us to understand or to visualize or to represent the interaction in between the protein and ligand molecules. So there are various approaches are there where we can check the visualization of docking ligand complex by using the various softwares, various tools. So here the PLP is there. Okay, so the PLP is considered to be as a protein ligand interaction profiler, protein plus, LigPlot. So these three different tools will give a different visualization pattern for us, like 3D figures, 2D figures, and you will see that some in-depth analysis like identification of cavities, and also you will find the two-dimensional structure. Also, it's very easy for us to understand what are the type of interactions are there. So apart from that, Pymol is also one of the option for us where we can visualize our protein and ligand complex. The major limitation with the Pymol is it will show you only hydrogen bond interaction for you. So it's not going to show you other non-covalent interactions. I'm going to show you some of the softwares for you where it will show you various kinds of interaction between the protein and ligand molecule. So in first step, what we need to do is we'll save the docking complex as a PDB format file. Okay, then we will start to visualize that protein. Okay, let's start. So the first step is we have to convert to the PDB format file. Okay, so we have to convert the DLG format into a PDB format file at the first step. So go to Autodock. So click on Analyze, Docking, Open. So here I save this one as the DLG format file. Okay, so DLG format file, open a DLG format file. Go to Analyze, Macromolecule, Open. So if you click on Open, it will automatically show you a ligand confirmation, protein ligand confirmation file. So again, go to here, Analyze, Confirmation, Play. So click on this AND option. Okay, so here show information. So you can see here it will show you a different type of information also for you. So as I told you, I just want a best confirmation. You can see here, so you will see the different confirmation is there. We already know the ninth run is the best confirmation. We got it, which is having a score of minus 14.17. Okay, so once you click on once you got this ninth run, if you got a doubt which run exactly you got the highest rank, go to DLG format file and check this one here, the number of runs. So it's the ninth one, you got 14.1. This is the highest binding free energy value. So here directly jump into the ninth one. Okay, so here click on write complex. Okay, you can save this one as a PDBQT format file. Initially you can see here, first we cannot keep this one as a PDB format file. First we have to save this one as a PDBQT format file. So I'm going to save this one as a PDBQT PQT. Okay, so here I save this one as a 1.pdbqt save. Okay, so now I saved this one. Okay, so you can visualize this 1.pdbqt in Pymol. If you go to Pymol here, click on this one, you can drag this one, you can visualize this protein and ligand molecule. But in some of the software, they are not going to accept this PDBQT format. That's why we have to change the format of this PDBQT to PDB format. So you can do this one in Open Babel. So here we have to first click on the PDBQT format file. Okay, so here give the path, and you can have to save this one as a protein data bank format file. So protein data bank format file output you can save as a best docking .pdb. Okay, so best docking .pdb, save on desktop, click on convert. Okay, so if you can't click on convert, automatically this all the components of this all coordinates has been written in the best docking format file. Now close this one. Now we have the PDB format file. Now this PDB format file can helpful for us to further visualization of your protein and ligand complex by using the various softwares. So let us start with the first one, the PLP, protein ligand interaction profiler. So in Google you have to type this protein ligand interaction profiler. So and also I'm going to give this web link in the description box. So here this protein ligand interaction profiler is a web service, fully automated. Okay, so you just need to provide a PDB structure, so whether your own PDB protein ligand complex file, or you can upload the already crystallized protein ligand by just giving the PDB ID to it. So here it will give you a complete list of non-covalent interaction in between the protein and ligand molecule. So let us start. So here I'm going to give PDB structure. Select this PDB. So choose best docking confirmation.pdb file. Okay, click on run. So click here small molecule. It will show you the name of that molecule and it will give a complete list for you. So here it will show you various kinds of interaction. You can see here not only it will show you image, and also it will show you tables also for you. What are the type of interactions are there in between the protein and ligand molecule? You can see here. So there are some hydrogen bonds also is there. They mentioned about what is the distance in between the hydrogen bond. You can see here 2.75 angstrom. The glutamic acid, alanine, arginine, serine, asparagine also is there, and also it will show you what are the donors and acceptor regions that are in the protein and ligand molecule region. So along with this, it will show you various kinds of interaction as I told you. So if it will show you a hydrogen bond interaction, hydrophobic contacts, pi size, stacking interaction, salt bridges, water bridges, halogen bonds, metal complex. It will show you so many things for you. So not only it will generate you high quality images, and also it will give us a Pymol session file to customize our PDB file, where we can change the style of the visualization style of the protein and ligand molecule. Okay, so you can save these images directly. Click here the PNG format file. If you want a PNG format file, you can save this one as a PNG format file. Okay, where you can see this image like this. And also if you want to save this table, it's very easy for you. You can just copy this table and paste in your Word or text document. Okay, so here If you want to save the Pymol session, you click on this Pymol PSC file. So here on desktop, open Pymol. So file, open, desktop, and best docking.psc file. Okay, you can see here it will show you a confirmation of protein and ligand molecule for you. So if you want to change this protein and ligand molecule as a stick format, we can change that information. Okay, so here go to sequence. So go to sequence here. So click on win the first drug molecule. Here preset ball and stick. Okay, you can see here. So it had shown in a ball and stick formation. So it will change this confirmation as a ball and stick for you. We can change the various confirmation, whatever the confirmation you want for your publication purpose. Okay, this is about the PLP. The next database we are going to check Protein Plus. Protein Plus server. So here also very simple one. You just need to provide your protein complex file. So choose best docking complex, go. So here in this Protein Plus, it not only generated 2D figures for you, and also you can perform the in-depth analysis in between the protein and ligand complex. You can see here visualization also one of the key here. You can change this confirmation here, the cartoon, whatever the confirmation you want, you can change here itself. White background, so whatever the background you want, you can change this information here. So and also you can the various options are available for you. You can check the hydrogen predictions, drug and ligand molecule binding score, 2D view, and also if you have a docking complex in between the protein and protein, you can check the protein protein interactions, and also you can see protein metal complex geometry. So if you want to see the water molecule placement, you can see, so we can do so many things by using this tool. So for our protocol, we are going to check the two things. One is a drug score. Okay, so click on do G site scorer. So you have to select the pocket. Okay, don't select the sub pockets. So here the pocket. So automatically, so we are going to check the drug ability of the ligand molecule. So if you want to check the properties also, there is option is available. So here in this option, we are going to select this drug ability. Click on calculate. So here this is the results we got. We're going to get this in the result form. Okay, if you click here in a show option, it will show you the where exactly your ligand has been, in which specific pocket the ligand has been interacted in the protein molecule. Okay, so it will provide you a surface area, the cavity, the pocket, a surface area in a protein, and also it will show you a drug scores also for you. If you want to know more about this one, I will just provide the link for you in the description bar about the DoG site scorer, how you can interpret this drug score. Okay, so along with this one, I'm going to show you one more thing for you here, post view 2D interaction diagram. So click on this one, click on post view. So here it will show you a ligand molecule. So it will ask you what are the ligand molecules are there. You just need to click here in this ligand molecule. So it will automatically load this name of this ligand molecule and click on the calculate. So here you can see the results. It will show you a hydrogen bond interaction along with the drug molecule in a 2D format. Okay, you can save this image in a PDF format or PNG format file. So click on this PNG format file. So here I'm going to save this one. You can check this protein and ligand interaction in 2D format file. This is how we can use this Protein Plus tool to visualize the 2D gram and also to calculate the drug likeness of your ligand molecule. Okay, so as I told you, you can use this all other options depending upon the type of analysis you're doing. You can use all this information. So next one is we are going to work on the another tool called as a LigPlot. So the LigPlot is from the EBI. Okay, so you have to download this software. So click on this LigPlot GUI format file and click on download. So once you've done this one, Ligand Plus, it is supported with the Java. You have to install the Java also in your system. Okay, so click on this Lig Plus. So click on the open PDB file. Okay, so you browse desktop here the PDB, select the PDB, click on best docking. Will show you what is the ligand inside your protein, inside your complex. Okay, select that one, click on run. So here it will show you 2D diagram along with the hydrogen bond interaction. You can see here there is a two hydrogen bond interactions are there in between the protein and ligand molecule, which distance is around 3.15 with the glutamic acid, and this 3.01 with the alanine molecule. So along with this, it will also show you a various amino acid cavities surrounding to this particular drug. You can change this one if you want to add a specific interaction in. For example, if you want to add a hydrophobic interaction, there is an option is available for you. You can click the hydrophobic and Hall's interactions of the protein and ligand molecule. So you can change the colors. So you can change the colors here. If the type of interaction you want, type of bond you want, you can do many things here. Once you've done this one, click on this write PS file. So here, post a script format. Save this one, the landscape, current selected prod. Okay, so click on save. Now it has been saved as a PS format file. You can you open that one in Adobe Photoshop or in any image formatting tool to save this one as a JPG or a PNG format file. So here the three tools are I showed you, where you can generate a 3D format files and 2D format files. So in Pymol, a few more options are available. If you want to work with the Pymol only, if you want to show only the hydrogen bond interaction, so then you can work with the Pymol as the best one. Also, okay, so you can drag any file, PDB format file or PDBQT file. So here I just drag this PDBQT file. First I just go to the Pymol action, preset, publication mode. Okay, so here now I selected this ligand molecule, click on this ligand molecule, here click preset balance stick. Now its representation is much better. If you represent in a balance stick matter, it will representation is much better for you. So here go to file, again the selected one, find polar context to any atom. So it will show you here the hydrogen bond interaction in between the protein and ligand molecules. So now you can see here we don't know which specific amino acids are there. So for this reason you have to go to show line. So select this one. So whatever the interacting regions are there, select the specific amino acids. Okay, so now I selected this one, click on hide lines. Now click selected regions, show sticks. Now you can see here, so now the protein ligand interaction is very clear for us. Okay, if you want to hide the carton molecule, if you want to show only the sticks and the drug molecule, just simply go here, hide cartoon. Now it will show you only interaction in between the amino acids and the ligand molecule. Now you can save this image if you want. You can see here, click on this file, export as image, PNG format file. Okay, you can save this one as a image. So you can change the background. You can check our previous Pymol lecture where you can change the background colors. You can annotate this amino acids also by adding different parameters like surface, and also if you want to add some mesh to it, you can do many things. These are the two things you definitely need for the publication. One is preparation of a docking results in a table format, and also the visualization part. Okay, so thanks for watching. If this video is helpful to you, please give a like, and if you have not yet subscribed, please subscribe to the channel for more amazing content in bioinformatics.